Hello and welcome to OSDR Chats. Today we have the opportunity to speak with Dr. Daniel Weiner and Dr. David Furman of Buck Institute. Using OSD 420, a dataset available from the Open Science Data Repository, these researchers and their team validated data from simulated microgravity experiments with spaceflight studies and reported potential countermeasures to maintain a healthy immune system during spaceflight. Today, Dr. Weiner will share a highlight of the study and its implications, and Dr. Furman will discuss how the Open Science Data Repository, or the analysis working groups, contributed to this publication. Thanks for joining us today, Dr. Weiner. Could you please highlight the most important findings in your study?
Well, thanks for having us. It's been known for a while that people who go into space, even in low Earth orbit, show compromised immune systems. Some of the common clinical manifestations of reduced immunity in space would be things like mild infectious diseases. This would be like, common colds. Astronauts also get skin rashes. There's also this reactivation of latent viruses, especially herpes viruses. These would be things like Epstein-Barr virus, varicella-zoster virus, cytomegalovirus, and this is seen in a large proportion of astronauts. So there is this immune dysfunction in space, but nobody really understands why. And that was the goal of our work, is to understand this immune dysfunction in space, and also to see if we can develop things that could help people in space, especially pertaining to the immune system. Since in low Earth orbit, microgravity is one of the dominant stressors on the body, we decided to map out in high detail how microgravity impacts the immune system. And so we did the first single-cell analysis of the human immune system in simulated microgravity with validation against spaceflight data. From this work, we identified key genes and pathways altered in immune cells at single-cell resolution by microgravity. And then we defined a number of core hallmarks of immune dysfunction linked to microgravity that reproduced among spaceflight data. Some of these pathways included microgravity effects on the cytoskeleton, interferon signaling, cell death, temperature shock, that would be heat shock, cold shock, and also changes in proteostasis, innate inflammation, nuclear receptors, sirtuin signaling, and mitochondrial dysfunction. We could then take these altered genes and, using machine learning algorithms, identify compounds that specifically reversed the detrimental effects of microgravity on the immune system. David and we like to call these compounds some of the first smart AI-developed space nutraceuticals, hopefully with potential one day to help maintain normal immunity in space.
Thanks for all the research that you're leading in this area. And my second question is for Dr. Furman. How did the Open Science Data Repository help assist you in your work so you could achieve these research goals?
Yeah, wonderful. Thank you, Jennifer, for having us here. So there are mainly two efforts that are bringing all the data from astronauts into one place, right? The one that we have been using for quite a while, and not only applies to astronauts, but also different animal models and plants and stuff, it's an Open Science for Life in Space Gene Lab. That's NASA, right? And we've been collaborating with them very closely to analyze different data sets from animal models as well from plants, and to see how different organisms react to space. The other very important piece that centralizes many of the data that we've been collecting, collaborating with Dr. Chris Mason at Cornell, is the Space Omics and Medical Atlas, SOMA. And this effort is really a collaborative project aiming to standardize biological measurements and data sharing across researchers for astronauts and spaceflight-related omics. So we're talking about proteomics, metabolomics, transcriptomics, microbiome, and stuff like that. And so using our established methods for analysis from the NASA twin study, the original twin study, we really are now expanding way more comprehensive and integrated multi-omics in astronauts, including the crew from Inspiration 4, and that is the SpaceX mission, Polaris Dawn, and Axiom missions. So looking at this longitudinal sampling before, after launch, so during spaceflight, and after return to Earth, we're able to monitor what happens at the level of biology in astronauts in the real life and how that impacts the health of our crew members.
It's great to hear the fruits of all your collaborations and how you've got all these connections now. We appreciate hearing from both of you about your insights and experience with the research. And thanks very much for sharing your time with our viewers.
Thank you so much.
And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access the data sets analyzed in this work with the links provided in the description section below. Thank you and have a great day.